Thank you to the CAR organization committee that gave me the opportunity to present the results of a portion of a collaborative Canadian postmortem study. I am a PhD student in Nicolas Chaumon's lab at the University of Montreal, and I'm presenting the characterization of HIV reservoir in multiple tissues. Also, I have no conflict of interest to declare. We know that people living with HIV need to take their ART regimen for life due to the presence of viral reservoir that will reactivate once the treatment is interrupted. The vast majority of reservoir studies on humans are performed on blood samples. However, we know that HIV can persist in different tissues. Studies, whether on monkey models or with access to human biopsies or autopsies, have revealed the persistence of HIV in many organs. The most studied ones are lymph nodes. But there's also a considerable amount of HIV DNA in the gut. HIV can also be found in the liver and the spleen of ART-treated people. The brain is another well-known anatomic reservoir. Some Canadian groups have also shown the implication of lungs and testes as HIV tissue reservoirs during ART. Identifying the tissues in which HIV persists during ART is a prerequisite to the development of efficient eradication strategies. Getting access to human deep tissues for HIV research purpose is achievable when people living with HIV generously decide to give their body to science at the end of their lives. The last GIVE study at the University of San Diego recently allowed to show that HIV reservoirs persist in all deep tissues and that blood and lymphoid tissues are the main source of dispersal. In Canada, the Medical Assistance in Dying law was adopted in June 2016. In 2018, 2 people living with HIV gave their body to HIV cure research here in Canada. The first individual was described by Jonathan Angel's team in a 2019 publication. He is a 67-year-old male from Ottawa, diagnosed HIV in 1987. He received medical assistance at death on March 16, 2018. He reported taking his medication until the day before his death and had no AIDS-related disease at that time. The second individual is a 68-year-old male from Edmonton, diagnosed in 2003. He died of B-cell lymphoma in June 12, 2018. He had several comorbidities, and his viral load was undetectable at the time of death. The objective of this portion of this collaborative study is to use 13 to 15 snap-frozen tissues collected post-mortem from these 2 participants to quantify and perform genotypic analysis of infected cells in multiple anatomical sites. Our hypothesis is that HIV persists at different levels in different organs, even in the context of ART. The objective here is to quantify both HIV DNA and RNA levels in the different tissues. This was performed by my colleague Julia Ruiz through qPCR of dual RNA DNA extracts. The other hypothesis is that intact, potentially replication-competent proviruses can be found in those tissues. This will be analyzed by doing near full-length proviral amplification from limiting dilution of DNA extract of the tissues, which is the part I performed. Total HIV DNA quantification was analyzed as followed. Each dot represents a single 3-replicate analysis of a piece of tissue. Values of HIV DNA is normalized on the CD3 level of the same sample. When no HIV was measured, we plotted as an open circle the limit of detection for that sample. Of note, we didn't have sample for neither hilar or axillary lymph nodes for Edmonton. For this part of the study, we see that in both individuals, HIV was measurable in all tissues analyzed. However, the highest HIV DNA levels were found in the different lymph node for Ottawa, while in Edmonton it was higher in the liver and the spleen. We also measured the cell-associated GAG RNA normalized on microgram of RNA of each sample. In the Ottawa participant, HIV transcript was detected at low levels, mostly in lymph nodes and lungs. However, a lot of samples were lower than the limit of detection. This can be explained by the fact that the autopsy was conducted 12 hours after death, which is a little bit late to preserve the quality of RNA. On the other hand, Spleen and lungs has the highest level of RNA measures for Edmonton. We then perform near-full-length amplification by nested PCR of the whole HIV genome and sequence the amplicons by PacBio technology. The amplicons cover the viral genome from the end of the 5' LTR to the end of the 3' LTR. The results presented here are not complete since other amplicons are in the process to be sequenced, but this was put on hold due to the COVID-19 measures. For Ottawa, we obtained 45 HIV proviral sequences so far from 9 tissues, ranging from 760 base pairs to 9 kb. We detected 10 proviruses that are over 8.9 kb of length, but all of them are defective due to deletions in the packaging signal or the presence of hypermutations. We also analyzed clonality of the proviruses. Clonality means we identified sequences having no more than one nucleotide difference. In the case of people undergoing effective ART regimen, clonal sequences suggest proliferation of an infected cell. Clonal sequences were found in multiple organs here. One clone is found in 3 different lymph nodes and in the lung. Another one is found in the axillary lymph node and in the rectum. And one clone is shared among hilar lymph node, spleen, and colon. We retrieved 43 proviral sequences from 7 tissues of the Edmonton participant. These sequences range from 1.2 to 9 kb. 7 out of 8 full-length proviruses are intact, 1 in the lung, 1 in the mediastinal lymph node, and all 5 spleen sequences. Interestingly, only 2 of these 5 intact sequences in the spleen are clonal. As for Ottawa, clonal sequences were observed in multiple tissues, but to a lesser extent. A big clone was found in multiple copies in mediastinal and inguinal lymph node, and a smaller one too. An intact clone is shared between lungs and spleen, and a short clone is detected both in the duodenum and the jejunum. Of note for this participant, all proviruses which contain the V3 region of the GP120 are X4 tropic. These results show that intact proviruses can be found in tissues, and that clonal sequences are shared in multiple tissues, suggesting proliferation of infected cells followed by migration to other sites. In summary, although detected in nearly all tissues analyzed, HIV DNA and RNA were more abundant in lymph nodes, spleen, lungs, and liver. The majority of proviruses were defective, as it is expected, and some clonally expanded genomes were detected in multiple organs. This tends to be different from publications describing compartmentalization of the HIV reservoir. However, we don't have enough sampling to properly characterize compartmentalization in this study. Finally, in one participant, intact proviruses were primarily detected in the spleen as secondary lymphoid organ that would need deeper studies. In conclusion, our study is the first one to perform near-full-length whole HIV genome sequencing of post-mortem tissues, combined to total HIV DNA and cell-associated RNA quantifications. It reveals a large diversity in the frequency of infected cells in different tissues, and highlights inter-individual differences in reservoir locations during long-term ART. This study increases the importance to get a better understanding of tissue reservoirs on ART. Since this is a collaborative study for HIV cure research in Canada, I would like to thank all the people involved. From my lab, a special thank to Julia, who started this project and performed the quantifications, and to all my colleagues. Thank you to the other students that performed the tissue collection, Amélie, Thomas, Saïm, Rosalie, Tesline, and Stephanie, and their lab directors. To the funding organization, especially to CanCure and CIHR, I would really like to thank both participants without whom we would have not been able to achieve this deep tissue HIV reservoir study, and thank you for your attention.